Yes. So carbohydrate biosynthesis in plants and bacteria. So carbon dioxide assimilation in photosynthetic organisms. Last week we talked about photophosphorylation, and that occurs during what cycle of the plant? Or it's called the what? Do you know? The light cycle. Questions like that are too simple?
No, I want to say photosynthesis, but I feel like I'm looking at this too simplistically.
Yeah, the light cycle, you know, is the capture of energy from the light, and you produce ATP, and what's the other thing that you produce during in the light cycle? Yes, NADPH, or reducing potential. That reducing potential and the ATP will be used in this cycle for carbon dioxide assimilation or carbon fixation to produce glucose, right? And it does this with the most abundant enzyme in nature, which is called Rubisco. It's the most abundant protein in nature is this enzyme, because there's so many plant life and algae and whatnot that does this photosynthetic pathway. So the previous chapters were mainly concerned with catabolism and how to extract energy from biomolecules. For this and the following chapters are concerned with anabolism, right? The building of things, or building of biomolecules. So you can always remember those, you know, catabolism and anabolism. You know, anabolic steroids are making guys bulk up. So anabolism, so building up. So plants are extremely versatile. They build organic compounds out of carbon dioxide, move intermediates between cellular compartments, use energy of sunlight to support this biosynthesis, and then they can also adapt to changing environments. And animal cells use 3 carbon intermediates, right? Pyruvate, lactate for synthesis. Plant cells will also make use of these 3 carbon intermediates for further synthesis. Glyceraldehyde 3-phosphate is an important one. And so that's the intermediate that will get from carbon fixation in order to go towards making sugar. So glyceraldehyde 3-phosphate is made from carbon dioxide, water, plus ADP, and your reducing potential from NADPH. That was produced during photosynthesis. Making carbon dioxide into intermediates is carbon dioxide assimilation, or carbon assimilation. We've heard that before, carbon fixation. So assimilation of carbon dioxide into the biomass. So we get the light cycle produces ATP, NADPH. That, along with carbon dioxide and water, will produce these triose phosphates, or glyceraldehyde 3-phosphate. Triose phosphates can be condensed to produce the hexose phosphates, which can then be used to make sucrose, or polysaccharide can be used to make starch for energy storage, or cellulose for structural. And then, of course, the pentose phosphates, right, are used to make DNA, RNA, proteins, and lipids also. So carbohydrate metabolism in plant cells versus animal cells. Glycolysis and gluconeogenesis are basically identical, right? It's a highly conserved pathway. Plants uniquely reduce carbon dioxide to the triose phosphates, such as the important 3-phosphoglycerate. Both plants and animals use covalent modification and allosteric effectors for regulation. So what's an allosteric effector? You guys remember what those are? Do you understand the meaning of the word? So where is that word? Allosteric. So allosteric could be in contrast to orthosteric. Have you ever heard of orthosteric, or ortho versus allo? No? So ortho means it's the same. Steric is like on the molecule, you know, steric interference. So orthosteric is like the same binding site. Allosteric is a different binding site. All right? So allosteric effectors, they bind to an enzyme or a protein, and they alter its affinity for its natural ligand, right? So allosteric effectors, one example is citrate, right, for the what's it, phosphofructokinase 1, which is the entryway into glycolysis. So citrate will inhibit that enzyme, right? And then there's the 2,6-bisphosphofructose. I don't know if I say that right. But remember, that's an allosteric effector for phosphofructokinase 1 also. So just same types of allosteric effectors for regulation that you see in plants that we have already discussed for animals. Plants also regulate the enzymes via reduction of a cysteine-cysteine cross-links via electron transport originating from photosystems 1, and then changes in pH and magnesium from illumination. So what do we mean by reduction of cysteine-cysteine cross-links? How do cysteines cross-link? Do you know? Do you remember? What's the functional group on that R group on a cysteine? It's often cross-linked in proteins. Do you guys remember? So it's the these things never work. Where's the garbage? This thing didn't work at all. It seems like I always grab them. Right? Do you remember the thiol groups? So when they say that you have reduction of cysteine-cysteine cross-links, cysteine-cysteine cross-links are these disulfide bonds, right? To reduce them, you just add hydrogen across, and you get, you basically break those cross-links with hydrogen, right? So you've reduced them. So that's all that means. And then if you reduce those cysteine-cysteine cross-links, it's going to alter, you know, the activity of that protein, because it's going to alter its rigidity and shape. So carbon dioxide assimilation occurs in the plastids. It's the organelles in plants and algae. Includes chloroplasts for photosynthesis and amyloplast for starch storage. So amyloplast, you've heard of amylopectin or amylose. Those are starches. Should make it easy to remember. Amyloplasts are for starch storage, if I could talk. Enclosed by a double membrane, just like the mitochondria, the inner membrane is impermeable to ions and polar molecules, and they have their own small genome, just like mitochondria also. So they can make their own proteins. But they still get proteins from the you know, that are transcribed by the nuclear DNA also, but they do make their own proteins, like mitochondria. Chloroplasts do the same. This is a photo showing you amyloplast filled with starch, and they stain these with iodine. The reason these stain so well with iodine is starch forms a helix that fits that iodine molecule perfectly, and when it binds there, it'll absorb light and give you this nice color. So types of plastids, there's undifferentiated protoplastids in plants. So proto, it's like pre-plastids. Can differentiate into chloroplasts for photosynthesis, amyloplasts for starch, chromoplasts for pigment storage, or elanoplasts for lipid storage, proteoplasts for protein storage. So it's the relationship between plastids. So don't worry about that. But we're going to focus on carbon dioxide assimilation. So it occurs in the stroma of chloroplasts via a cyclic process known as the Calvin cycle, right? Key intermediates. The key intermediate is this one, ribulose 1,5-bisphosphate. So it's constantly regenerated using the energy of ATP. So this is a 5-carbon sugar, right? So it produces 3-phosphoglycerate and then 3-glyceraldehyde phosphate. And which is equivalent with dihydroxyacetone phosphate, right? And then we use glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Those will condense to form fructose if we're doing gluconeogenesis, right? So the net results is the reduction of carbon dioxide with NADPH that was generated in the light reaction. So we use the NADPH, reduce the carbon in carbon dioxide, will produce water and sugar. So the overall reaction is takes 3 carbon dioxides plus 3 ribulose 1,5-bisphosphates is going to yield 6 3-phosphoglycerates. So those 6 3-phosphoglycerates can be used to make, they're then reduced to 6 triose phosphates, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. 5 of the 6 glyceraldehyde 3-phosphates are used to regenerate ribulose 1,5-bisphosphate, and the remaining 1 glyceraldehyde 3-phosphate is used to make the hexoses, for sucrose, starch, etc. So the overall reactions, 3 carbon dioxides, 6 NADPHs plus 5 waters, 9 ATPs produces glyceraldehyde 3-phosphate, 2 hydrogen ions, NADP, and 9 ADP plus 8 phosphate, free phosphates. And then there's 3 stages in the Calvin cycle. First is the carbon fixation. So it takes 3 ribulose 1,5-bisphosphates plus 3 carbon dioxides is going to yield phosphoglycerate. And then that's catalyzed by that enzyme I was telling you about, Rubisco. So Rubisco catalyzes the reaction where you fix carbon from ribulose into ribulose 1,5-bisphosphate. Step 2, 3-phosphoglycerate is reduced to glyceraldehyde 3-phosphate using NADPH and ATP from photosynthesis, and then catalyzed by 3-phosphoglycerate kinase and glyceraldehyde 3-phosphate dehydrogenase. Regeneration of ribulose 1,5-bisphosphate completes the series of reactions. And this is how they go. Stage 1, fixation. We take in the carbon dioxide. It gets added to the ribulose 1,5-bisphosphate, and you produce 3-phosphoglycerate. ATP comes in, so does NADPH, and you form the glyceraldehyde 3-phosphate then. That can be used for energy production, and then you have to regenerate this ribulose 1,5-bisphosphate afterwards also, and that takes more steps, and we'll see how that goes. So Rubisco is short for ribulose 1,5-bisphosphate carboxylase oxygenase. So we'll just call it Rubisco. It's a large magnesium enzyme, so it uses magnesium as a cofactor, and it catalyzes the conversion of ribulose 1,5-bisphosphate to 3-phosphoglycerate using carbon dioxide. So as a carboxylase, it incorporates inorganic carbon dioxide into organic ribulose 1,5-bisphosphate, which is the first step of the Calvin cycle. It makes a new carbon-carbon bond between that carbon dioxide and 1,5-bisphosphate. And it also cleaves the 6-carbon intermediate into 2 3-phosphoglycerates. So you've taken a 5-carbon sugar, added another carbon, so it's now 6-carbon intermediate, and it gets cleaved into 2 3-phosphoglycerates. Can also react with oxygen in an inefficient side reaction, but we'll focus on the main reaction. So from form, Rubisco exists in 2 major forms, and it's the most abundant enzyme in nature. Form 1 we find in plants, algae, cyanobacteria. It's got 8 large catalytic subunits, and it's encoded by the plastid genome. 8 small subunits encoded by the nucleus. So here's where we're getting 2 different proteins that come together from the, different subunits that come together from the same, that come together to form the same enzyme, but they're from the different genomes. One's from the plastid genome, the other's from the nuclear genome of the plant. So form 2, photosynthetic bacteria only produce, and it's only 2 catalytic subunits, which resembles the plant subunits. Low turnover number of 3 per second at 25 degrees C means lots of energy is needed to do this. 50% of plant enzymes are Rubisco. So, like I said, the most abundant enzyme in nature is Rubisco. So, this is a picture of the enzyme. So, this will be on the test. You'll have to draw this. No, just joking. But here at the active site, this is form 2, you can see there's magnesium. And then there's a modified lysine residue. It's carbamoyl lysine, so it has a carbamoyl modification. This shows you an up-close picture of that structure. You have the carbamoyl lysine. There's ribulose, 1,5-bisphosphate, carbon dioxide, glutamic acid, and aspartic acid. And the carbamoyl lysine kind of coordinate with that magnesium, and it draws in the carbon dioxide. And the ribulose 1,5-bisphosphate, you can tell these are the phosphates down here, are all attracted to that positive charge on the magnesium. The magnesium brings the carbamylated lysine to the ribulose 1,5-bisphosphate and forms an enediolate. Magnesium polarizes carbon dioxide to accept nucleophilic attack from the enediolate, and then you form this 6-carbon beta-keto acid intermediate. You get hydroxylation at the carbon number 3, cleavage into a 1,3-phosphoglycerate. The second cleavage reaction forms the second 3-phosphoglycerate. And we can look at these mechanisms in more detail. Let's see, how do I get over there? Again, so there's the magnesium, glutamic acid, aspartic acid, and then the carbamylated. They all coordinate with that magnesium. A carbamylated pulls off the hydrogen ion, and you form that enediolate intermediate. There's the ene group. And then this is primed for attack by these electrons under the carbon dioxide to form a new carboxylic acid group. Now you have a beta keto acid intermediate. Is that beta? It's not. I forget my Greek letters. It's a square keto acid, or not square, but a rectangular keto acid intermediate, right? Oops, wrong button. Water then attacks that carbonyl. You have a hydrated intermediate because you've added water across there. Electrons drop down from that, and then these electrons have to go somewhere. You form a carbanion intermediate, and then this here is your 3-phosphoglycerate, right? And then you have this carbanion intermediate. Picks up hydrogen ion from the lysine. And that's your second 3-phosphoglycerate formed from that ribulose in the carbon dioxide. Where is it? Okay. Catalytic role for the magnesium is it's held by the negative charge side chains of the glutamic acid, aspartic acid, and that carbamylated lysine. So magnesium brings together the reactants in a correct orientation and stabilizes all the negative charge. that forms upon the nucleophilic attack of that enediolate to carbon dioxide. So it's highly regulated as the first step of carbon dioxide assimilation. It's inactive until the lysine 201 is carbamylated by carbon dioxide. So that lysine 201 is inaccessible until it's carbamylated, right? And that's, you need that carbamylation. Rubisco activase is an enzyme that's sometimes triggered by light, changes Rubisco conformation to expose the lysine, allowing it to be carbamylated, and that requires ATP. So that should be easy to remember. Rubisco activase is the enzyme that will actually activate Rubisco by exposing that lysine 201, allowing it carbamylation. So this is how that looks. So how Rubisco activase exposes lysine 201. So you have Rubisco here, this lysine's not carbamylated. Unmodified lysine 201 and bound ribulose 1,5-bisphosphates, inactive. Rubisco activase uses ATP. Ribulose 1,5-bisphosphate leaves. ATP dependent removal of the ribulose 1,5-bisphosphate uncovers that epsilon amino group lysine, which will then allow the carbamylation and the magnesium to come in. So Rubisco is also inhibited by a nocturnal inhibitor, 2-carboxy-arabinanitol 1-phosphate. Inhibits the carbamylated Rubisco. It's the transition state analog of a beta-keto acid intermediate. So that's what the TS stands for. Synthesized in the dark in some plants. So, and this is the carboxy-arabinanitol 1-phosphate. So it's a transition state analog. If you guys remember from when we covered enzymes, they preferentially bind to the transition state. Transition state analogs are typically competitive inhibitors, so it's going to bind to that active state with high affinity because it's analogous to the transition state, but you can't catalyze the reaction with it. So stage 2, the 3-phosphoglycerate reduced to glyceraldehyde 3-phosphate. You have the 6-3-phosphoglycerate, 6 ATPs, 6 NADPHs, and 6 hydrogen ions yield 6 glyceraldehyde 3-phosphates. 6 ADPs, 6 NADPs, and phosphate. Requires NADPH and ATP from photosynthesis. It's the reversal of the glycolysis reaction, except NADPH is used for that reducing power rather than NADH. Uses enzymes 3-phosphoglycerate kinase and glyceraldehyde 3-phosphate dehydrogenase running in reverse. Driven forward by the high concentration of NADPH and ATP in the chloroplastoma. So 5 of the 6 are recycled, you know, the glyceraldehyde 3-phosphates. 5 out of 6 of those are recycled back to ribulose 1,5-bisphosphate. The remaining one can be converted to starch in the chloroplast or can be converted to sucrose in the cytosol for export. And this is stage 2, conversion of 3-phosphoglycerate to glyceraldehyde 3-phosphate. And where do we start with that? We have 3-phosphoglycerate. 3-phosphoglycerate kinase phosphorylates that to 1,3-bisphosphoglycerate. Then the NADPH will come in and reduce that to glyceraldehyde 3-phosphate. And then triosisomerase can convert one of these to dihydroxyacetone phosphate. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate can then be, you know, aldolase can convert both of those to fructose 1,6-bisphosphate. And then if you remember, fructose 1,6-bisphosphatase removes one of the phosphates or hydrolyzes one of the phosphates and produces fructose 6-phosphate, which ultimately can be used to make starch or glucose. One of these And then one glucose can then be used to make sucrose, but you have to be exported to the cytosol here for that step. The 3 carbon sugars are exported here, and then they can be used to make fructose and sucrose. Or can be burned as glycolysis also. So regeneration of ribulose 1,5-bisphosphate. It's a complicated series of reactions with 3 carbon, 4 carbon, 5 carbon, 6 carbon, and 7 carbon intermediates. And it generates a pentose phosphate pathway intermediates also. And the steps are, you get glyceraldehyde 3-phosphates converted to DHAP by triose phosphate isomerase. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are converted to fructose 6-phosphate, which is a 6 carbon via aldolase, and fructose 1,6-bisphosphatase, then removes that phosphate, right? 2 carbons are removed from fructose 6-phosphate to yield erythrose 4-phosphate, which is a 4 carbon intermediate. It's catalyzed by transketolase. Transketolase transfers the 2 carbons from step 3 to glyceraldehyde 3-phosphate, yielding Cellulose 5-phosphate, which is a 5 carbon intermediate. So these are all these crazy intermediates to produce ribulose 1,5-bisphosphate. And then we have erythrose 4-phosphate dihydroxyacetone phosphate. So we'll combine then to form cetoheptulose 1,7-bisphosphate, 7 carbon intermediate by aldolase. Cetoheptulose 1,7-bisphosphate is dephosphorylated to cetoheptulose 7-phosphate via cetoheptulose 1,7-bisphosphatase. And then this enzyme is unique to plants. Cetoheptulose 7-phosphate has 2 carbons removed by transketolase to yield ribose 5-phosphate. The 2 carbons on transketolase are transferred to glyceraldehyde 3-phosphate to yield another Cellulose 5-phosphate. And then ribulose 5-phosphates converted to ribulose 5-phosphate. And then by phosphopentose isomerase. And then also step 7 is the XU5P, Cellulose, right? It converts to ribulose phosphate by phosphopentose epimerase. And then finally ribulose 5-phosphate is phosphorylated to ribulose 1,5-bisphosphate by phosphoribulokinase. And this enzyme is also unique to plants. And this is the overall steps here. So it's kind of convoluted process to regenerate our ribulose 1,5-bisphosphate. So transketolase catalyzed reactions, we take the ketose donor and aldose acceptor, and you just switch that around, transketolase. You just donated the keto to the aldose, producing a new aldose and a new ketose. Fructose 6-phosphate and glyceraldehyde 3-phosphate are then used to make eutherose 4-phosphate and Cellulose 5-phosphate. So an example of another transketolase reaction, right? So we've taken the ketose from fructose 6-phosphate and donated it to the glyceraldehyde 3-phosphate, or switched from the fructose to the glyceraldehyde, producing Cellulose 5-phosphate, and eutherose is the remaining part of the fructose that was there. And then other transketolase reactions include cetoheptulose. Here we have the ketose that gets transferred to the glyceraldehyde 3-phosphate to produce another Cellulose 5-phosphate and a ribose 5-phosphate. So So TPP is the cofactor for transketolase. Do you guys remember TPP? So it's that thiamine pyrophosphate. What's special about TPP? And again, transketolase uses the thiamine pyrophosphate as its cofactor. Contains thiazolium anion for nucleophilic attack. So this is that carbon, it's the thiazolium anion. The hydrogen here is acidic, so it stabilizes this anion. Also used by pyruvate dehydrogenase, pyruvate decarboxylase, and alpha-ketoglutarate dehydrogenase. Transketolase in the pentose phosphate pathway also. So stoichiometry and energy cost of carbon dioxide assimilation. So we fixed 3 carbon dioxides to yield 1 glyceraldehyde 3-phosphate. So that makes sense, right? 3 carbons to make 1 3-carbon sugar. 9 ATPs and 6 NADPH molecules are consumed. And then the light reactions of photosynthesis produce ATP and NADPH at the same ratio, 3:2. So it's kind of perfectly matched, which makes sense that they'd have evolved like that. So stoichiometry, and this is the figure. 3 carbon dioxides produces the 3 phosphoglycerates, and then 6 ATP, 6 NADPHs produces 1 glyceraldehyde 3-phosphate. So the fate of phosphate from ATP hydrolysis in stage 1, 8 of the 9 phosphates combined with ADP to regenerate ATP. The 9th is incorporated in the glyceraldehyde 3-phosphate and requires that phosphate to be transferred from the cytosol, which uses a special phosphate triose antiport. So what does that tell you if it's a phosphotriose antiport? Phosphate goes one way, triose goes the other way, right? So the phosphate triose antiporter is needed for sucrose synthesis, right? So unlike starch, starch is made in the chloroplast stroma, sucrose is made in the cytosol, used for transport to distant plant tissues, but the inner chloroplast membrane is impermeable again to phosphorylated compounds. So the antiporter exchanges 3-phosphoglyceraldehyde 3-phosphate or dihydroxyacetone phosphate for 1 phosphate, thus sending the triose phosphates to the cytosol for sucrose synthesis. So exchanges with the phosphate that way. And this is the picture showing you that. So in the cytosol is over here, stroma of the chloroplast here. We have glyceraldehyde 3-phosphate and dihydroxyacetone phosphate can be transported out while phosphates transported in. Dihydroxyacetone phosphate can then be converted again to glyceraldehyde 3-phosphate, be reduced to 1,3-bisphosphoglycerate, and then phosphoglycerate kinase. 3-phosphoglycerate can be transported in again, phosphate as the phosphate goes out, so it's an antiporter. So photosynthesis from light and carbon dioxide to glyceraldehyde 3-phosphate. The photosynthesis of 1 molecule of glyceraldehyde 3-phosphate requires the capture of roughly 24 photons of light. The hydrogen move from the stroma to the thalkaloid, which creates alkaline conditions in the stroma. It's accompanied by magnesium transport from the thalkaloid to the stroma. The enzyme for photosynthesis and carbon assimilation are more active in the alkaline, high magnesium conditions of the stroma. So this is how hydrogen ions and magnesium movement assist the activation of photosynthesis and carbon assimilation. Little mouse. Where? Oh, right there. It is Mickey Mouse. So this should make you happy about photosynthesis. So And this shows you the fructose bisphosphatase 1 activity and how it works better if you see in more alkaline pHs than at neutral pHs and at magnesium concentrations. You need less magnesium to get this fructose bisphosphatase activity going than at pH 8 than you would at pH 7. Light activates 4 enzymes via electron-driven reduction of cysteine-cysteine crosslinks, remember? So the target enzymes are ribulose 5-phosphate kinase, fructose 1,6-bisphosphatase, and celluloheptose 1,7-bisphosphatase, glyceraldehyde 3-phosphate dehydrogenase. So if oxidized, cysteine residues have a disulfide form, the enzymes are inactive. So in light, the photosystem sends electrons to ferredoxin, which sends them to the thioredoxin, which will donate those electrons to the disulfide bonds to reduce them to the free cysteines. And that gives that enzyme more, I guess it's a less rigid state, more relaxed state. Remember the relaxed and the tense forms of enzymes? Disulfide bridge would form like a tense form, which makes it inactive. If you break those disulfide bridges, it's going to be in a more relaxed, more flexible state, which is a more active state for the enzyme. And this shows you that flow of electrons. So we acquire the electrons, get boosted up from photosystem 1 when you get absorption of a photon. It reduces FD, ferrodioxin, right? Reduced. Those electrons go to break this disulfide bond on thioredoxin. Then these electrons can break the disulfide bond on the enzyme, activating that enzyme, and then we produce oxygen in the dark from that. So mitochondrial respiration and photorespiration. So far we've seen that the plants oxidize water to oxygen and reduce carbon dioxide to carbohydrates, right? So that's what we do for photosynthesis and carbon assimilation. Plants also carry out the reverse in the mitochondria, where oxygen's oxidized to water, whereas substrates are reduced to carbon dioxide, just like we do, right? In addition, a wasteful side reaction catalyzed by Rubisco occurs in the mitochondria that consumes oxygen and yields carbon dioxide. This photorespiration reaction is driven by light and is coupled with a costly salvage pathway. Unlike mitochondrial respiration, does not yield energy. That's this oxygenase activity. So oxygen will compete with carbon dioxide for the active site. 1 in every 3 or 4 turnovers, you'll get oxygen binding rather than carbon dioxide. The reactive nucleophile in the Rubisco reaction is the electron-rich ene-diol form of ribulose 1,5-bisphosphate. The nucleophile adds oxygen to form 3-phosphoglycerate, same as in the Calvin cycle, and 2-phosphoglycerate. 2-phosphoglycerate is metabolically useless, so you have to salvage its carbon, and that is going to require energy to salvage that carbon. So the oxygenase activity of Rubisco. So again, we have ribulose 1,5-bisphosphate, the ene-diol form of that, and then oxygen will come in. You'll have this enzyme-bound intermediate, and you'll form 2-phosphoglycolate and 3-phosphoglycerate. And the 2-phosphoglycolate then needs salvaged. So the complex ATP consuming process for the recovery of the 2 carbon fragments from photorespiration involves the oxidation of the glycolate with oxygen in the peroxisomes, which will form hydrogen peroxide. Loss of carbon is carbon dioxide by mitochondrial decarboxylation of glycine. So it uses a glycine decarboxylase complex, similar to the pyruvate decarboxylase complex, and alpha ketoglutarate dehydrogenase. Right? Do you guys remember that big, massive complex? Ultimately, you get two phosphoglycerates, two phosphoglycolates are converted to serine and carbon dioxide, and that the oxygen is consumed and carbon dioxide is produced. Energy is required, not produced from this cycle. And this is the glycolate pathways. So we get the glycolate from the side reaction, gets out of the chloroplast. Glycolate acid oxidase forms glyoxylate, and that'll come. You have transamination from the serine to the glycine. Then that glycine will go to the mitochondria, where it'll be oxidized to form glycine decarboxylase, then releases carbon dioxide and produces serine, which can then come out and, get transamination again, produce hydroxypyruvate to glycerate, and that glycerate can then go back into the chloroplast and used for sugar production or ribulose 1,5-bisphosphate production. And if you remember these processes with the pyruvate decarboxylase or alpha ketoglutarate dehydrogenase, you have the PLP, the lipoic acid is that disulfide bridge, and your FAD. So glycine comes in, binds to the PLP, you get decarboxylation, then the lipoic acid picks up that intermediate, right? Then folate will come in and remove that. The folate comes in, you end up with methylene folate, so you picked up a methyl group and you've lost this ammonia group also. And then this lipoic acid needs to be reoxidized, so the FADH picks up the lipoic acid, then NADH can come in and re-reduce or reoxidize. What are we reoxidizing here? Oh, so NADH picks up the electrons from the FADH too. Just like pyruvate decarboxylase. Glycine decarboxylase is abundant in the photosynthetic parts of plants, and you have flux through Rubisco reaction, and the glycolate pathway can be high in bright sun. So plants need lots of the glycine decarboxylase, and makes up to 50% protein in some plant leaves. But non-photosynthetic parts of the plant, such as tubers, have little glycine decarboxylase there. So you get that a lot where you have photosynthetic and bright sunlight. Most plants cannot avoid oxygen binding to Rubisco. So if you have an atmosphere of 21% oxygen and 0.038% carbon dioxide, pure water has a 260 micromolar oxygen and 11 micromolar carbon dioxide at equilibrium room temperature. So we got way more oxygen exposure than we do carbon dioxide exposure. So the Km for Rubisco for oxygen is about 350 micromolar, and so you're below the Km here, right? But not too far below the Km. As discussed below, some plants, you ever heard of the C4 plants? Do they talk about that in core yet? And the CAM plants have a bypass mechanism to avoid this side reaction. So most of our crop plants are C3. As discussed, the first step of the Calvin cycle is CO2 fixation and do a 3-carbon product. The C4 plants have an earlier step before Rubisco, and it bypasses the C3 fixation by fixing carbon dioxide into a 4-carbon compound, oxaloacetate, from acetyl-CoA. Tend to grow in hotter, sunnier climates and have a high rates of growth, photosynthesis, low water loss, and a special leaf structure. And sugarcane, corn, maize, and sorghum are all C4 plants. So the C4 plants physically separate carbon dioxide capture from the Rubisco reaction. So carbon dioxide capture into oxaloacetate in mesophyll cells of the leaf. Oxaloacetate is derived from acetyl-CoA. Oxaloacetate can then pass into bundle sheath cells and release that carbon dioxide. In the bundle sheath, carbon dioxide concentrations then much greater than oxygen concentrations, and this is where Rubisco is located. So you don't have so much of that oxygen binding rather than carbon dioxide. So the C4 plants often outperform C3 plants where you have heat and drought. C4 pathway has higher energy costs, but as increased temperature and affinity for Rubisco for carbon dioxide, gain in efficiency which outweighs that energetic cost. Hence the C4 plants, some are weeds, sometimes do better in heat and drought than the C3 plants do, because they have that payoff there overall. And this shows you the different parts where you get carbon assimilation, the mesophyll cell and the bundle sheath cell. This is the overall C4 pathway. So in the mesophyll cell, you get pyruvate, gets converted to phosphoenolpyruvate, and then PEP carboxylase will take that bicarbonate and produce oxaloacetate. So we've taken the carbon from carbon dioxide here, fixed it into oxaloacetate from phosphoenolpyruvate. Oxaloacetate can then be reduced to malate and transported into the bundle sheath cell where malate gets reoxidized, decarboxylated to pyruvate, and then the carbon dioxide can combine with ribulose 1,5-bisphosphate to form the 3-phosphoglycerate, and you get the normal carbon assimilation there. So CAM plants separate carbon dioxide trapping and fixation in time. Another pathway to avoid the Rubisco reaction with oxygen. The Rubisco reaction with oxygen? Is that a typo? Sorry. First discovered in crassulaceae, so-called crassulaceae acid metabolism, or the CAM cycle, found in plants that grow at high temperatures and dry conditions. And this is pineapple, cacti, and minimized loss of water vapor. CAM plants open gas absorbing stomata only at night. So at night the air is cooler, more moist. Carbon dioxide absorbed at night is fixed to make oxaloacetate via the PEP carboxykinase reaction. Oxaloacetate is reduced to malate and stored in the vacuoles then. So in the daytime, the malate releases carbon dioxide via that NADP-linked malic enzyme, and carbon dioxide fixation proceeds. Since the stromata are closed, oxygen is low then. So mid-chapter summary. Huh? Yeah. So ATP and NADPH from the light reactions needed to assimilate carbon to carbohydrates in the dark reactions. Assimilation of 3 carbon dioxides via the Calvin cycle leads to the formation of 1 molecule of 3-phosphoglycerate. 3-phosphoglycerate is a precursor for the synthesis of larger carbohydrates such as fructose and starch. The key enzymes of the Calvin cycle, Rubisco, fixes carbon dioxide into the carbon dioxide, and the low selectivity of Rubisco causes that wasteful incorporation of oxygen. And this is avoided in C4 plants by increasing the concentration of carbon dioxide near the Rubisco, and in the CAM plants by storing carbon dioxide in malate and releasing it, when oxygen is not being absorbed. So another 75 to go. No, I'm just joking. So excess triose phosphate is then converted into starch and sucrose. Starch is made in the chloroplast for short-term storage, or made in the amyloplast of non-photosynthetic tubers, seeds, roots for long-term storage. So that's, potatoes are starchy. Say long-term storage, they store that energy in the tubers. So sucrose made in the cytosol for transport. Starch provides the bulk of energy storage for most plants, except for sugar beets, sugar cane, which they use sugar to, sucrose to store that. It's all sugar. This is making me hungry. So starch synthesis requires activated glucose 1-phosphate. But unlike glycogen, glucose monomers in starch come from ADP glucose. If you guys remember from glycogen synthesis, it's UDP glucose. So amylose has all alpha 1 to 4 glycosidic bonds. Do you guys remember how those are? Amylopectin has alpha 1 to 6 branches added, by a branching enzyme, similar to glycogen, but amylopectin doesn't have nearly as many branches as glycogen has. So, synthesis of sucrose uses UDP glucose, so more like us. Sucrose has alpha 1 to 2 bonds, so it's the, alpha 1 from the glucose to 2 of the fructose, which makes it a non-reducing sugar. This is the anomeric carbon of glucose ends up in a glycosidic bond with the anomeric carbon of fructose, and that makes it a non-reducing sugar because you don't have that free carbonyl form of either of the sugars. They're both fixed in that ring form when they're in this alpha 1 to 2 bond. So to be synthesized in the cytosol, triose phosphate precursor must be imported from the chloroplast, and we already saw that that's done via that triose phosphate antiporter. So sucrose synthesis involves the conversion of dihydroxyacetone phosphate, glycerol aldehyde 3-phosphate to fructose, followed by addition of UDP glucose. So aldolase combines with dihydroxyacetone phosphate 3, glyceraldehyde 3-phosphate to form fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate is dephosphorylated again by fructose 1,6 bisphosphatase, just like in gluconeogenesis, to form fructose 6-phosphate, and then sucrose 6-phosphate synthase adds the UDP glucose to the fructose 6-phosphate to yield that sucrose 6-phosphate. Sucrose 6-phosphate is then, cleaves that phosphate to yield just plain old sucrose. So this is the overall steps, right? So UDP glucose and fructose 6-phosphate forms sucrose 6-phosphate. Sucrose 6-phosphate phosphatase removes that phosphate, and you're left with sucrose, which is that sweet sugar we like in our food, right? Or add it to our coffee or whatever. Regulation of sucrose synthesis occurs at interconversion step of fructose 1,6 bisphosphate and fructose 6-phosphate, right? So this is similar again to gluconeogenesis, right? Fructose bisphosphatase 1 and PP fructose phosphofructokinase 1 have the reverse reactions. Pyrophosphate dependent phosphofructokinase 1. So similar to the glycolytic enzyme. So the forward enzyme fructose 1,6 bisphosphatase, we've heard that before. That happens in gluconeogenesis, just removes that phosphate to form fructose 6-phosphate. And then for the reverse reaction in plants, it's pyrophosphate dependent phosphofructokinase 1. So So in the dark, no photosynthesis occurs, no triose phosphates, and no sucrose synthesis, but sucrose breakdown is needed. So fructose bisphosphatase 1 is inhibited, right? Because we don't want to make more sucrose or spend energy making sucrose. We need to convert it the other way, basically like activating glycolysis. We're going to pyrophosphate phosphofructokinase 1 is active, and in the light reactions, the reverse is triose phosphates are made. So we're going to synthesize sucrose. The fructose bisphosphatase 1 is active, whereas pyrophosphate phosphofructokinase 1 is inhibited. So simple regulation there. So you want to remember that fructose 2,6 bisphosphate is an important glycolysis gluconeogenesis regulator in animal cells, made and broken down by that bifunctional enzyme made from fructose 6-phosphate by phosphofructokinase 2 activity, favors glycolysis, sucrose breakdown, broken down by fructose bisphosphatase 1 activity, favors gluconeogenesis or sucrose synthesis. So during photosynthesis, triophosphates are made, phosphofructokinase 2 is inhibited, fructose bisphosphatase 1 is active, and that favors sucrose synthesis. So fructose 2,6 bisphosphate as an allosteric regulator of that sucrose system. So in the dark reactions, we activate pyrophosphate phosphofructokinase 1, which will convert it to fructose 1,6 bisphosphate, which makes it to triose phosphate, which is the direction of glycolysis. In the dark reactions, we do the reverse, right? Fructose bisphosphatase is blocked in the dark, rather, and we go this way towards glycolysis. Fructose, where are we? So dark reactions, phosphate will leave into the cytosol and activate phosphofructokinase 2, and at the same time that blocks the fructose bisphosphatase 2 activity. Do you guys remember the production of fructose 2,6 bisphosphate for glycolysis? How that regulates phosphofructokinase 1? Same thing in plants, kind of. So sucrose 6-phosphate synthase is also highly regulated. It's activated by glucose 6-phosphate. Glucose 6-phosphate also inhibits the kinase that makes sucrose 6-phosphate synthase. Sucrose precursors are abundant, so that'll make sucrose. Sucrose phosphate synthase is inhibited by phosphate, so the phosphate also inhibits the phosphatase that makes sucrose 6-phosphate synthase more active. Photosynthesis is too slow to make sucrose. So regulation of sucrose phosphate synthesis by phosphorylation here. And then starch synthesis, did I say that wrong? Went the wrong direction. Starch synthesis is regulated at ADP glucose pyrophosphorylase. It's activated by 3-phosphoglycerate. So if you have lots of 3-phosphoglycerate, you want to store those as either sucrose, convert them to sucrose, or store them as starch. It's inhibited by phosphate. Phosphate is going to accumulate when the condensation of ADP and phosphate slows. So you're not making as many ATPs, so phosphate will accumulate. So rather than making sugars and storing them, you're going to start burning sugars to make more ATP, right? And that regulation. This is slow in the dark, right? We're not using ADP to make ATP. So we have an excess of phosphate, which is going to block this ADP glucose pyrophosphorylase. and we'll shift it to glucose 1-phosphate and ATP. And we won't be making the ADP glucose and pyrophosphate, which is used to form the starch. Cell walls are strong due to cellulose, right? So more than 10 to the 11 metric tons made per year of cellulose. So that's a lot of carbon fixing, right? And a lot of structure, a lot of wood. Glucose monomers with beta 1 to 4 linkages make starch. Straight shape allows those chains to cross-link via hydrogen bonds, so you form those really strong fibers in the cellulose. Synthesized from intracellular precursors but assembled outside the plasma membrane. And this shows you that microfibril, the beta 1 to 4 linkages of glucose that form that starch. And you form interchain and intrachain hydrogen bonds with all the alcohols on the sugar. So the steps in one model of cellulose synthesis: glucose attaches to the lipid in the cell membrane. Sucrose synthase generates UDP glucose, and with the help of cellulose synthase, 6 chains are created. The cellulose protein complexes form rosettes. You can see those on electron microscopy. Cellulose synthase extends the chain. Growth laying down of the polymer appears to be directed by the microtubules, and the chains crystallize to form fibrils. I can show you these, the EM rosettes here, where you're getting the active cellulose synthesis. And the bacterial cell walls are made of peptidoglycans, because remember from micro. And they use intracellular precursors to create that extracellular structure. Cell walls are made from alternating linear polymers of 1-acetyl-glucosamine and N-acetyl muriamic acid, also linked by beta 1 to 4 bonds, just like cellulose. And then they're cross-linked by peptides to hold those together. The pentaglycine cross-link holding those together. So plants can also use fats and proteins for energy, just like animals. Plants store lipids and proteins in seeds. Gluconeogenesis provides glucose plus intermediates for sucrose, and then used in germination, growth, process unique to plant cells and some microorganisms, converts acetyl-CoA to glucose. Remember, animals cannot convert acetyl-CoA to glucose, only plants and microorganisms. So animal cells make glucose from 3-carbon lactate, glycerol, 4-carbon oxaloacetate precursors, but plant cells have enzymes that are able to convert acetyl-CoA into glucose. Breakdown from fatty acids occurs largely in the glyoxysomes. The glyoxylate cycle of chapter 16 generates succinate sent to the mitochondria, converted to oxaloacetate, and then glucose. And this is where you get that. So you have glyoxylate cycle produces succinate, goes in the mitochondria to produce oxaloacetate, and oxaloacetate decarboxylates to phosphoenolpyruvate, and then that just follows normal gluconeogenesis to produce glucose 6-phosphate, or glucose ultimately, which can be combined with fructose 6-phosphate to form sucrose. The glycerol from stored fat in seeds can be used to convert to glucose also. Glycerol moiety can be converted to dihydroxyacetone phosphate and then glycerol 3-phosphate to enter the gluconeogenic pathway, similar to us. So remember, from fats, triacylglycerol. The fatty acids can be used to produce acetyl-CoA via beta oxidation. The glycerol via glycerol kinase is produced glycerol 3-phosphate, and then glycerol 3-phosphate dehydrogenase oxidizes it to dihydroxyacetone phosphate, which is one of the 3 carbon sugars used to make fructose or glucose. And that's it for this